Blood transfusion is a very important part of modern medicine. A blood transfusion may be necessary after a serious accident, during or after a major operation, or following childbirth. Sometimes an exchange transfusion is necessary in a newborn baby because of blood group incompatibility. Blood transfusion is the most successful tissue transplant. It was first carried out in 1818, but sometimes a severe or even fatal reaction occurred. It was not until 1900 that Landsteiner discovered that people have different blood groups. Because of this, we need to match the blood of the donor with that of the recipient. The main blood group system is the ABO system. The entire human population can be divided into 4 groups on the basis of the reaction between the blood of different individuals when mixed together. The reaction is immunological, causing normal red blood cells like these to clump together or agglutinate. This is agglutination. It is what happens inside a patient if they are transfused with blood of the wrong group. It can obviously have very serious consequences for the patient who's already ill. There is a constant need for blood donors. An adult human has about 5 litres of blood. When half a litre of blood is donated, the body soon replaces it. Since blood doesn't keep all that long, regular donations are essential. Surgeons cannot embark on major operations unless they know that ample supplies of blood are available from the blood transfusion service. Giving blood is safe and painless, and of course helps others and saves lives. It is important, however, that the donor doesn't have any infectious diseases, especially hepatitis or AIDS. After the donation is completed, a sample of blood is taken. This will be tested to establish whether or not it contains any infectious agents. Human blood is classified into 4 main groups: O, A, B, or AB, depending on the presence of certain glycoproteins or antigens on the surface of the red cells. Group O cells only carry a backbone molecule called H. Groups A and B have extra sugars on the H backbone molecule. Group AB has both the A and B molecules on the backbone. These molecules are called red cell antigens. They are not antigenic in the person they came from, but if they are injected into a person who does not have them, they will stimulate antibody production. High levels of antibodies to blood group antigens seem to exist spontaneously in all blood groups except in people of group AB. For example, the group O person's immune system will recognize A and B in red cells. People who are blood group O, therefore, have anti-A and anti-B antibodies. People who have only A red cells have anti-B antibodies, and people who have only B red cells have anti-A antibodies. Here is how the blood grouping is carried out in the laboratory. We need samples of known anti-A and anti-B antibodies. In fact, these days we use monoclonal anti-A and anti-B antibodies as standard laboratory reagents. The reagents are colour-coded to avoid the possibility of picking up the wrong bottle. The anti-A antibody contains a blue dye, and the anti-B antibody contains a yellow dye. We also include known control blood samples in the test: A, B and O, to be sure that the test is working properly. A small sample of the donated blood is put into a labelled tube. This sample is then diluted with saline to make the reaction easier to see. The used pipette must be disposed of safely. Remember, it may be contaminated with HIV or the hepatitis B virus or some other infectious agent. When full, the container will be sealed and sent for incineration. The blood grouping is carried out on a white tile divided into 4. Two drops of each blood sample, A, B, O and test, are added to the appropriate square on the tile. A new pipette is used for each sample, and the used ones are disposed of in the sin bin. Next, 1 drop each of anti-A and anti-B antibody is added to each square. The blood is mixed with the antibody solution. Notice that a new mixer is taken for each sample to avoid cross-contamination. The tile is then rocked gently to mix the solutions thoroughly. In a minute or so, clumps start to appear in the positive samples. The results are clearly visible to the naked eye. The test blood in this case was group AB. Down the microscope, the individual red cells can be seen to have agglutinated. Here is the result of another test with group O blood. Remember that O individuals, who comprise a major proportion of the population of Great Britain, do not have either type of antigen in their blood, so their red cells will not clump with anti-A or anti-B. Although the ABO blood group system is the main one, there are a number of others that need to be matched if a reaction to blood transfusion is to be avoided. The most well-known of these is the rhesus system, under which individuals are grouped as Rh positive or Rh negative. Human red cells are routinely typed for the presence or absence of the rhesus antigen, also known as D antigen. This antigen occurs independently of the ABO antigens, so individuals of any ABO type may be Rh positive or Rh negative. Rh negative blood can be safely transfused into any individual with the appropriate ABO group. But Rh positive blood given to an Rh negative individual would stimulate antibodies to the Rh antigen and result in a transfusion reaction.
Hands are a little bit cold.
The rhesus, or D antigen, can also cause major problems in pregnancy.
Right. And is this your first baby?
It is.
Let us assume that a rhesus negative mother is carrying her first rhesus positive child. When the baby is born, some of its blood may enter the mother's circulation. The mother's immune system recognizes the baby's red cells as foreign and makes antibodies to the rhesus antigen. If the mother becomes pregnant with a second rhesus-positive child, the mother's anti-rhesus antibodies may destroy the fetus's red cells before birth. This may result in hemolytic disease of the newborn, causing jaundice, which can be fatal unless the child is given an exchange transfusion. This problem can usually be prevented by treating the mother with anti-rhesus antibodies within 1 day of her first delivery. This destroys any fetal cells that get into her circulation, preventing the sensitization of the mother's immune system. Because of the existence of a fairly large number of blood group systems in humans, blood transfusion centres carry out extensive cross-matching before allowing blood to be used for transfusions. The National Blood Transfusion Service depends entirely on voluntary donations. Whole blood is given when there is blood loss as a result of an accident or during a major operation. In addition, many components of blood are separated and stored for future use. There is a constant need for blood donors. You can get further information by ringing this number.